I would like to briefly explain our GUT paper published in 2020. Iqgap3-Ras axis drives stem cell proliferation in the stomach corpus during the homeostasis and repair. What has been known on the subject? Isthmus of the stomach corpus is highly proliferative and considered to be the place where stem cells are located. Cytoskeletal protein Iqgap3 is necessary and sufficient for cell proliferation. This was published by other group. So what are the new findings? We identified Iqgap3 as a functionally important stem cell factor of the isthmus stem cells. And Iqgap3 is expressed in actively proliferating cancer cells of virtually all types of cells, which are shown in this slide. We identified Iqgap3 as a stem cell factor in the isthmus. Rapidly proliferating stem cells are located in the isthmus of the corpus in homeostasis. Iqgap3 is co-expressed with the Ki67 in the isthmus. Furthermore, Iqgap3 is co-expressed with the Ki67 in majority of the gastric cancer cells. But not only that, according to publicly available database, Iqgap3 is expressed in probably all types of cancer. Iqgap3 is an embryonic gene and re-expressed in cancer cells. If it is knocked down in cancer cells, they are induced to differentiate. So this means Iqgap3 is required for cancer cell phenotype, which also means that this is probably the potential drug target. So in this graph, we show the knockdown of Iqgap3 in NTERA2, which is human embryonal carcinoma cells. When we knock down Iqgap3, NanoOcto4, KLL4, CMYC, SOX2, these genes are all well known to be stem cell factor. The expression of these genes are strongly reduced. At the same time, GFAP, which is a differentiation marker of this cell line, is strongly induced. That is clearly shows that after knockdown of Iqgap3, cells are induced to differentiate, and therefore it is important to maintain the phenotype of stem cells. So high-dose tamoxifen induces tissue damage in the corpus of the stomach. This is because destruction of the parietal cells. High-dose tamoxifen probably induces the expression of Iqgap3 in terminally differentiated cells. In this slide, it is shown as a Ki67 positive cells, but in this case, Ki67 positivity and Iqgap3 expression are virtually identical. This initiates tissue repair process. Gene expression patterns during this process shows a similarity to early-stage gastric cancer, as you can see on the right panels. So, our observation is consistent with the notion that tumors, wounds that do not heal. This is proposed by Dvorak in 1986. So, conclusion. Iqgap3 is required in stem cells in homeostasis, tissue repair, and cancer. Although Iqgap3 is cytoskeletal protein, it has a profound effect on maintaining the phenotype of cancer cells, probably all types of cancer. Thank you.